Alright, today we are going to talk about IA. We look at the checklist in more detail and double check whether everything given in our lab report is following the checklist or not. So the first point given here is topic. So you have to identify the topic which is the part of your syllabus of your investigation. So for example, enzyme, protein, digestion, immune system, whatever, you just identify the topic and you can give the title along with the topic, that for example, effect of temperature on enzyme. So if you have given this title, it includes enzyme, we understand, okay, topic is given. It no need to be more detailed, but a precise one sentence title or a topic with one word is sufficient enough along with the title. Second point, RQ. So RQ needs to be relevant, focused, and clear, and it should be clearly described. So it means RQ should include the independent variable, dependent variable, as it is written in the lower part. RQ includes independent variable, dependent variable, species, or the name of the material used. How it is collected, it means the raw data. If you have given a time, should be given how long you have put the setup, what process is happening. So if it is a osmosis or it is aerobic respiration or anaerobic respiration or digestion, should be clearly given. So what is the example? Does different concentration of hydrogen peroxide affects the rate of oxygen produced in 5 minutes when the solution is put with blended potato containing catalase. Now in this one, this is a research question mark should be there. I have given the independent variable, I have given the dependent variable, which is the rate of oxygen get produced in 5 minutes. What is the material? Potato. You can give the scientific name in bracket, which should be in italic. Containing catalase. So this one is the enzyme is here. It means the potato contains catalase. Now one thing which I could put it here, how it is measured, by using oxygen gas, oxygen sensor, or whatever way you are going to use. Measured how? Using oxygen sensor, or use the inverted measuring cylinder technique, or you use the gas syringe. So if I'm putting all this information, my RQ is very clear and all the information is described in the RQ. Now surprisingly, some of you have not put the question mark in the RQ. And another mistake is you are starting your RQ to what extent where there is no t-test, it means you are not comparing. If you are not comparing, you should not start your RQ with to what extent. So RQ should be clearly relevant, focus described clearly. Now RQ is relevant, it includes IVDP and species name. These two parts comes in the RQ. Point 4, background information is appropriate, relevant, and enhance the understanding of context of the investigation. Now, background information, some of you have written introduction before it, where you are telling how, what ignite you or what stimulates you to research on this question. It's fine, but if you have given the heading personal engagement, this is a wrong heading. You are not supposed to give a separate personal engagement heading, because it is the overall report which give you marks, not that one paragraph for personal engagement. So in the introduction, you can give the significance and why you reached to this research question, but definitely should not be given a heading of personal engagement. Background information should include all information which is supported by theory with all referencing. If I'm giving the enzyme as an example and defining enzyme, this should be referenced. Then this investigation, if I take this investigation as an example, so enzyme, I talk about catalase. I will talk about if the concentration of hydrogen peroxide, what is the use of hydrogen peroxide, what hydrogen peroxide is doing here. And this information I'm getting from some resource, and that should be referenced. Then I'm using potato. So what potato I'm using? What is the theory or the enzymatic theory or biological information about this the potato I'm using? It also should be referenced. So everything here should be referenced. If I'm using the oxygen sensor, how the oxygen sensor works. If I'm using some other apparatus, which is, for example, I'm using spectro or refractro or whatever apparatus I'm using, how it is working, and it should be referenced, because this is not your information. So background information is very important to be very clear to get good marks in exploration, because here we get the idea that how well the student understand the report. All right, now moving on to the next part, again reiterating, it should be reference. Every information should be referenced. Hypothesis is quantitative, and a sketched graph with explanation is included. This hardly any of you have done it. Hypothesis, as suggested, you should be write down it as prediction as the heading. But if I'm hypothesizing something, my hypothesis is coming based on my background information. If I'm talking about the concentration, higher the concentration, more the rate of reaction. So it should be, for example, if my concentration is 1%, 2%, 3%, 4%, 5%, and 6%, my prediction is lowest at 1%. Don't write down this way, I'm just explaining you, okay? Lowest at this, highest, followed by this, this, and this. So all the concentrations should be mentioned in prediction, not only high and low. and this is the concentration prediction is only based on the information you have given in the background information. Now predicted graph. Now if I'm making a predicted graph, I should reference it. For example, if it is a simple investigation of concentration like this or temperature, you can easily get the graph online and you can reference it. But if you are not getting anything and you are making it, you can make by yourself using software, and you have to tell the source of the software used. All right. So your hypothesis and prediction must include the quantitative, the number should be there, and a sketch graph is more relevant if it is possible. Now coming to point 6, IV is identify by detail of range. Now I found very few of you are providing me clear details. I need clear details. You identify IV as the concentration of hydrogen peroxide. What is the effect of concentration? You talking about it here, but how you are getting this concentration should come here. So the range of 1%, 2%, 3%, are you getting it? How are you making it? If you have given me the independent variable as temperature, and the temperature is, you have given me range 1, 2, 3, 4, 5, are you using water bath? How you controlling it? Using water bath, electronic water bath, or the Bunsen burner, you have to mention. Or if you have taken like some your investigation in on a type of fruits, and you have a range of 5 type of fruits, you have to tell how you are controlling it by taking a same mass or same volume or the brand. Every detail should be here. Now details of dependent variable. The dependent variable, again, if you are calculating the rate of enzyme reaction, you have to mention here how you are calculating the rate very, very clearly. So the oxygen gas O2 produced in 20 minutes recorded by how oxygen gas sensor and all, and then you are to calculate rate. This is divided by whatever time you have fixed it over time. So this is how you're calculating the rate. So this clarity of how you are collecting your dependent variable, how you're getting your dependent variable should be very clearly mentioned here. Next is your control variable. Same thing. For example, in this you are taking the type of food used, type of food which I'm using, what is you're going to explain why if you're using different one. And then I said potato, the scientific name is given here, and then the volume. If you have to, you can't just simply say okay, I'm going to control the volume. How is it, how much, and how means if it is 20 cm cube, how did you control? By using measuring cylinder. Did you use 10 cm cube measuring cylinder or 20 cm cube measuring cylinder, or did you use pipette? Should be mentioned here. You can't just leave your answer in the lab. You can't just leave this place that okay, it is standard volume. How much is this and how did you measure should be clearly mentioned. Okay, the setups left for example, another many of you have not given me precise details here of the control variable. Many critical control variables are missing, which you should double check with your procedure. Check your procedure. Whatever numbers are given should come in your control variable. Temperature, you are saying temperature was controlled. How it is controlled? Are you using water bath? Are you using aircon? What temperature are you keeping your water bath at? What temperature are you keeping your aircon at? Should be clearly mentioned here. Thermometer, if your independent variable is temperature, use of thermometer to monitor is must. So control variable, list of control variables should be very clearly given, that how you are controlling, not only what you are controlling. All right, so DV detail uncertain means it is written here. Hardly any of you have given the unit with uncertainty in the dependent variable. So please use this checklist given to you. Control variable, identify each control impact the result, how each will be control value with method for achieving that value should be clearly mentioned. Variable that could not be controlled are identified and method for monitoring included. So for in this example, you know that you are taking potato, but if you are not taking if you are not taking all the samples from one potato and you have to take two potatoes, you have to use two potatoes, you cannot control the exact nature of two materials, so that is uncontrolled. But you can probably say that you took it from the center or you took from the same batch, so you try to make it standard. So uncontrolled variable are those which you definitely cannot control, but you try to make it standard, so make it a precise answer. Okay, so we are done with half of it. 9 parts are done. So you will get this video with half, otherwise the video will be very long and I'll making another one in a short while. Please make use of it, because I'm sitting on Sunday and making